Hello everyone, my name is MV Yashwant Rahul and today I am presenting a project titled Java's Game Representation for Biological Sequence Comparisons. This work is focused on a unique visual and computational approach to analyze the DNA sequences using fractals and machine learning. Now we discuss about abstract Java's Game Representation. Java's Game Representation or CGR is a powerful mathematical visualization technique or powerful mathematical visual technique. It allows us to transform complex biological sequences into intricate fractal images, unlike traditional sequences alignment methods that really dwell heavily on string matching. CGR helps in graphical methods, and in this project, I have used CGR to visualize DNA sequences mapped and nucleated bases to corners of square of AGCT and generated fractal images. These images were analyzed using machine learning, specifically random forest model to classify DNA sequences based on features like intensity and shape. Here is my content flow of my presentation: introduction, objectives, background, method, CGR visualization, machine learning approach, current status, results, and that outputs. I will show that summary and references what from where we have taken. In introduction style, Java's game representative was introduced as a tool for sequence analysis with the power of fractal geometry. In biology, DNA and RNA and protein sequences hold the key to biological functions and disease pathways. Traditionally, we analyze these sequences using alignment based to leukoblast, which are computationally expensive. Java's game representation brings a new angle by transforming sequences data into fractal images, enabling visualization pattern detection. We have shown that further. I will show that this production aims to explore how CGR helps us to compare classified DNA sequence more effectively. The main objectives of this project is the objectives of slide is visualize biological sequences, compare sequences based on CGR patterns, leveraging machine learning for classification, enhance genomic research. It contributes to genomic research, contributes to improve tools for genomic research for diagnostics, mainly in hospitals and research centers they will use. And here we apply machine learning on CGR for classification knowing. And is visualize output for CGR to identify structural similarities between different organisms and species. In background slide, sequence alignments methods are effective but time consuming, so may not detect all differences. So CGR offers visualization method wherever sequences become images with unique patterns. Fractals, which are self-repeated patterns, help capture structural and biological complexity. This background sets the foundation applying CGR in bioinformatics. So traditional methods like blast and needleman wounds and aligned sequences are calculated similarity based on exact matches or gaps. These methods can help slow computational expensive large data sets will help you. It helps. And a fractal geometry plays a significant role in visualizing hidden where data is hidden. It might be known. So in methods I have mentioned here CGR mapping, sequence processing, feature extraction, fractal dimension, color intensity, area, classification. So classification only means random forest I have used so that I have mentioned classification. So in this method, each DNA sequence A and C and G and T is assigned a corner of a square. So starting at the center and the algorithm moves halfway towards the corner corresponding to each nucleotide. This iterative process from a fractal images after that statistical features such as fractal dimension and pixel intensity are extracted. So these features are used to train the machine learning model to classify sequences based on visual similarities. So here I mentioned that only for instance, it might be top left corner G and G at the top right, C at the bottom left and T at the bottom. So sequence processing starting at the center of the square, we reach each nucleotide in the sequence, move half of the towards the corner, and this will repeat the recursive process. So once the CGR is generated, various statistical features will be shown that it will be dots. I will show in that in output that the picture will be dots and like that. Color intensity reflects the nucleate frequency and sequence characteristics. I have successfully complemented the small dataset of DNA sequences, and at this stage on CGR, I have used a dataset for sample DNA sequence to generate CGR images. Next, I extracted a key features and trained random forest classifier. Initial results are promising with good classification accuracy. Challenges include scaling to model large datasets for refining for better results. So, further steps are expanding the dataset to include more sequences from different organisms, extracting techniques, and exploring advantages of machine learning models like deep learning. I will try because to improve sequence classification. Here the what we did the codes all I mentioned. So CGR CGR generator and main method I have shown here, and we have used FASTA files from different species and shown CGR fractal outputs, classification outputs, and here you can see CGR output different DNA sequences. Each pattern is unique, visually represents the structure of the sequence. So I will show you. Here you can see the CGR output of different DNA sequences. Each pattern is unique and visually represent the structure. As seen, these visual patterns can be directly usable for data machine learning. The classifier trained able to differentiate sequences effective. So the results suggest that a CGR is an effective alternative to traditional sequence alignment. The random forest classifier is able to group similar sequences based on their fractal features with over 85% accuracy on our test. So this confirms that even simple CGR features carry biological meaning. In the future, we plan to use CNNs and deep learning features to improve the classification accuracy and generalization. So this is the mutation to mutation. So the summary is this slide, this project what we did up to now. CGR transforms DNA into fractals that carry structural information. This project combines fractal visualization with machine learning to develop a tool for comparing and classifying sequences. With further improvements, CGR could be scalable. Visual and methods bioinformatics pipelines for large scale DNA analysis. So initial results suggest that CGR is an effective method for sequence analyzing. With further improvements expanded as more data is analyzed, the project holds potential for improving bioinformatics tools used in genomic research and medical diagnostics. So this is the summary of the slide. This is the references I have taken from the DML lesson and Jeffy, Jeche, and Almia. I have here pasted the links. I refer to the keywords in fractal geometry, bioinformatics, and machine learning. These includes books we have mentioned, journal of CGR in biology and research articles. So thank you for watching my presentation. I hope this give a clear understanding of how Java's game representation can use DNA sequence comparison and classification. Thank you.